Hello, dear fellows. Welcome back to the channel. And we have just started a few topics of chemistry, and we have studied a couple of topics for A levels related to the IGCSE, a few topics related to adactyl chemistry. And now today we have just started the lectures for the biology, which will cover different aspects of it. And in today's discussion, our focus will be on straight on the basic concepts of biology, just that engage the DNA. So we're going to start it with the DNA. You have to be clear, like DNA is something which is called as double-stranded structure. It's just like a stair-like structure, or maybe the letters who are having a gum-like structure in which them like the steps are engaged to hold the two strands of DNA in the simplest way. There is a ladder, and among them, the steps are basically built up by the hydrogen bonds. Okay, so DNA, as per its introduction, is the deoxyribose nucleic acid in which oxygen at the carbon number 2 is absent. So that's it. It is some sort of, it lacks oxygen at carbon number 2. It's the only difference between the like RNA and DNA. And other difference is of the uracil. So these are the two major differences. But firstly, we will focus on the thing like it lack oxygen was supposed to be present at carbon number 2 in the RNA, but in DNA, they lack this oxygen. Now, as for this introduction, all the living cells comprise of more than one type of cells, which are basic units of their structure and function. The concept is like all the living cells, even within a human body, every cell have its own DNA, and in that DNA, a specific gene is enabled while it does contain genes for other stuff or other activities, but they are in the muted form. So if I compare the genes of your liver, even the genes of your smell and the genes of your taste, so both of them contain like your tongue also contains genes or the makeup for the smelling activity, but by the nature they have been muted. So your tongue is only able to taste the food in the same way your nose is bound to smell the stuff. So in a simple way, every cell contain every makeup, but they are muted. They are inactive to do other activities. So the simplest way is like all the living cell comprise of more than one cell, and every cell have its own DNA. That DNA is enabling every cell to perform various activity that it have to do. Okay, now DNA could be termed as the genetic material. So what this genetic material is like, it is basically your life controller. In that perspective, we could simply say that when you're born or whenever a baby just starts its growth, that is only a cell or maybe the two. So as soon as the time passed by, the activity which is called as mitosis, which we will study later on, which is going on, which starts when you were born or when you're in your mother's womb. It just started from that phase, and it just kept on going until a human dies off. So simply, it is a life controller in the two different perspective or the broad terminologies, which is called as mitosis and meiosis. So whenever the reproduction occurs and a baby is about to produce the fusion of egg and sperm cells, they give rise to something which is called, the activity engaged in it is called as mitosis. Sorry, meiosis. But when the cells are going to multiply, and as soon as the growth proceeds and the organism is getting bigger and bigger in their sizes, that is uh, aided by mitosis. So that is simply when cell divides from 1 into 2, 2 to 4, 4 to 16, and that is how they're going to complete their growth. So during your entire life, both activities goes on, but majorly it is life is controlled by the mitosis. So that is also aided by the DNA. The next point is like transfer of the genetic information. It is simply the point or the concept when DNA is transferred from in an inherited pattern from parents to their offspring. And remember that the DNA or simply the DNA contains a chromosome, or simply the DNA is built up by the chromosome that is in a cross position. So that way So among that cross, one thing, one side, one chromatid, this is the thing, is donated by your parents, and this is by the newborn, this is by the offspring zone. So this thing is responsible, or what we could call it as the transfer of information from parents to their offspring. So that is the reason, like child or offspring have similar characteristics to that of their parents, maybe in their physical appearance, maybe their different like characteristics. So that is the way it is the transfer of the genetic information from one generation to another generation. The next point is like DNA structure. The structure of DNA is simple, as we have just like discussed already. So simply, this is the thing. Even there is the thing. So DNA structure is like this way. That is how DNA is arranged. And this stuff is called as the hydrogen bonding. So these are the nucleotides, like there multiple nucleotides like adenine, guanine, thymine, cytosine. They are engaged in building up of the genetic material, and now those nucleotides are held together, or they are bonded together via the hydrogen bonds. That hydrogen bonds number vary from the group to group, or from the like they vary, like when they have to make bond between the adenine and thymine, they make two hydrogen bonds. When it comes to the bonding between the guanine and cytosine, they make three hydrogen bonds among them. So in that perspective, they are like holding different nucleotides together. So simply, nucleotides are polynucleotides, basically like lots of nucleotides are bonded together. They are holded together by the majorly by hydrogen bonds, and some role of the covalent bond is also responsible to hold them together. Now the building blocks are nucleotides, and basically these are the like we need nucleotides to give rise to a huge DNA structure. So their building blocks are the nucleotides. So as per their structure, every nucleotide must contain three groups. It must contain a sugar group, a phosphate group, and nitrogenous base. So simply, that is the simple structure of the DNA, or simply the nucleotide. So here is the five carbon structure, and like there, it contains five carbons. Every corner is represent a single carbon. So right now there are five carbons. They have one phosphate group. One phosphate group is attached to four oxygen atoms along with one hydroxyl group. So we will discuss it later on. It also contains one nucleotide base, like adenine is also available there. Okay, so as per detail, like a five carbon sugar is available. So in the simplest way, the pentose in DNA is deoxyribose. That is same as that of the ribose, but the difference among them is like they on the carbon number two, it lacks one oxygen atom, while in this case in ribose it contains an oxygen. So that is how it is the major difference among them, and that has changed the entire nucleic acid classification, like for the DNA and RNA. Okay, so deoxy word represents like it lacks oxygen onto it. So how you will study the structure of DNA? So simply that contain one oxygen, one carbon there, right there, this carbon one, two, three, four, and ultimately carbon number five is there. So what we need, or what would be our major focus is like carbon number three and carbon number five. Okay, so I guess you would be clear till this point. The next point is the nitrogenous bases. So these nitrogenous bases are simply held together by the covalent bond, and the bond is termed as phosphodiester bond, or the linkages. So these phosphodiester bonds are responsible for holding the nitrogen bases together. Now phosphate residue is attached to the hydroxyl group of the fifth carbon sugar of the one nucleotide, and the hydroxyl of the 3' carbon of the sugar of the next nucleotide. So they give you 3 to 5 phosphodiester bond. With this statement, I mean to say that this red thing is called as phosphate group, and that is the point. Like this point, there would be an OH group present. So, and right there at this fifth carbon, there was a hydrogen bond available. So it is an OH string. So simply, this phosphodiester who was supposed to be like this way, like P along with its OH, and that P came, or phosphate came along with its OH group that came, and it attacks on that OH group of the fifth carbon, or 5' carbon. It just threw up this thing and it just dragged its hydrogen out of it, and water molecule just release off. So simply that could be called as condensation. After donating its hydrogen and taking, after donating its OH group and taking hydrogen from the fifth carbon, it just released a water molecule. Okay, so that is called as phospho phospho diester bond. Now, why I am calling it as diester bond? It's like one ester bond is produced right here. The second ester bond is going to produce like this position, like the carbon number 3. Another phosphate group would came and it is going to attack on this thing, and it would drag its water OH out of it, and they would be releasing another water molecule. So that is how two phosphate groups are going to attach onto it by releasing water molecules, and we can simply say that they are releasing upon every attachment of phosphate group. They had released two water molecules. So simply, whenever two phosphate groups are going to attach with this nitrogenous base, they will release one hydrogen upon each attachment. So ultimately, 2H2O would be released upon attachment of two phosphate groups. Okay, now at this point, it connects to this thing. This side is responsible for connection of nitrogenous bases. So that gives rise to these nitrogenous bases. So they are going to attach on this like carbon number, on the carbon number 1, and that is how they are going to establish the DNA structure. Now, the DNA structures are of the two types. So one is the pyrimidine ring, and other is the purine ring. So you should be clear with the difference among them. Like when it comes to the pyrimidine, one ring is responsive. One ring, they contain one ring, and when it comes to the purine, the two rings are like joined together like that way. You can see this one, but when you compare these rings to this side, like cytosine or thymine, one ring is there. So simply that is the major difference. Pyrimidine consists of one ring, while pyrimidine consists of two rings joined together. Okay, so the next point is like when we have phosphate group. Now that phosphate group is responsible for DNA double helix structure. It is going to provide support to it. So there you can see it is going to attach different sugar with each other by providing them with the support. So that is how they will be attaching different different sugars along with each other. DNA consists of the two strands, and obviously they are winding like a winded up in a twist-like structure. That twist is supported by the phosphate groups. So that is how the three basic structures aid in the building up of the DNA. The next point is that DNA is present in the three forms. What we have studied is the B form of the DNA, but the DNA exists in the two other forms. Even there are five major forms of DNA, but generally the three models or the three forms are majorly accepted. Okay, so that is the form number, form A, form B, and form Z. So what is the basic feature of the DNA is the helix, like this way. So that is the helix when the two strands are strongly twisted upon each other, and the two strands are wrapped upon each other, you could say that. Now we have heard the two terms. One is the major groove, other is the minor groove. So when you see like the DNA consists of like this is having a structure of like this way. So okay, so that's all. Now that side which is a bit thin and covering less surface area, this is the minor groove, while this thing is termed as the major groove. So that is how, and obviously the major groove contains, it is a broad, covering the broad area, it is having bigger inside, so that is how the DNA is present on. Okay, so DNA, what we have been studying is like basically the DNA, the B form of the DNA. Now, what's happening there is B form is available enough, is available commonly. Form A is the one who is and the who is having right-handed turns onto it. they are covering most of they are twisted at the right side. So basically, the nucleotide basics are like turned or bind towards the axis, which is the right hand side. So basically, you can see it is tilted towards right hand side. So we could say that it is the right-handed DNA or form of the DNA. So when it comes to the B form of the DNA, they are also right-handed DNA like this way. They're highly hydrated because yeah, they are highly hydrated. But when it comes to the form A, it is although less hydrated, but it is more compact. While when it comes to the zDNA, that zDNA is tilted more towards the left side. So that is how it is arranged. It is tilted more towards left side. So we could say that it is left side of the left-handed side of the DNA. So that's it for its the general introduction. Now for another discussion, DNA contains lots of histone bases or histone on it, histone protein on it, sorry. That histone is making it negatively charged. So, okay, so that's it. Now, the forms of DNA for the summary, we have studied three forms of the DNA, A, B, Z. Form A and B are both are right-handed side, right-handed, while when it comes to the Z, Z is left-handed, and side A or form A is less hydrated, but the best thing is like it is more compact, while the side or form B is highly hydrated and it is right-handed, it having a right-handed turn onto it. Okay, the next one is like each strand serve as a template. With the template, we mean like it having a strand like this way. Now on this strand, it contains some bases onto it like adenine, guanine, cytosine, and thymine. Now it would be wandering here and there in search of another template who will be containing the complementary strands to it, just like that should contain this T. For G, it must contain C. For C, it should contain G, and for A again, it contains the A. So simply, that template is looking onto the complementary strands. So in that way, it contains each strand of DNA would be serving as a template or a pattern to make a new DNA molecule. So if you just broke these strands just like that way, they are containing some opposite sequences onto them and opposite bases onto them. So in order to make the bonding into them, if you just broke them, they would be looking upon some sort of alternative bonding. So we have as just initial level here studied E is going to make a bonding with T and it is bound to make two bonds with it. While when it comes to the guanine, it should be making three bonds with the cytosine. So in that way, it is going to act as a template looking for the complementary sequence so that it could find them and make the new DNA sequences. The next one is helicase enzyme. Helicase enzyme is what? Remember that enzymes are basically the 3D structures who are going to accelerate the reaction without being participating into them. So simply they speed up the rate of reaction, but they are not going to be used up in that reaction. So in case of helicase enzymes, helicase enzyme is responsible for breaking the hydrogen bonds between the bases that holds the two strands together. Remember that when you need it, you just need that helicase enzyme when you have to stop the bonding. So whenever something needs a protein and you have produced and your body has produced enough protein and it doesn't need any more protein, the helicase enzymes just came, it just cut on the sequences so that the side that was needing protein and your body was producing protein, it could stop that protein formation. So that was just an idea to like when you need that helicase enzyme. Also, it is responsible for unzipping the DNA. So whenever you need to stop an activity to go ahead, you are bound to unzip the DNA, and obviously you need helicase enzyme to break the hydrogen bonds. So the next point is like DNA is unzipping when nucleotides attach to their base pair by the hydrogen bond, and when you broke those hydrogen bonds, it is called the unzipping of the DNA. Another thing is like DNA polymerase enzyme. It binds the nucleotides into the chain, so its role is inverse to the helicase enzyme. Helicase have to break the bonds, and DNA polymerase have to attach the nucleotide bases to give rise to the lengthy DNA, and you need it when it comes to the translation, the transcription, and you need helicase enzyme when you have to end up the transcription and translation. So that activity continues until the whole molecule is unzipped and replicated. Remember that you need these enzymes to build up the long chain structure, to build up the DNA, to and obviously you need them when it comes to the stopping the DNA. That is role of DNA replication. That is the role of helicase enzyme. So simply that was it for today. And whenever certain changes occur, like enzymes got failure to do its activity as it have to do, like the normal activity, something which is called as mutation occur. Mutation is what when DNA or anything, the DNA just stop doing its normal activity, it behaves in an abnormal way. That is the one example of mutation. So we will cover it in the next class. We will cover it in the next class. So that's it for today. As per summary, we have discussed about the DNA. In a simplest way, we have compared the two things, ribose and the DNA. Ribose is when they lack or they contain oxygen on the carbon number two. When it comes to DNA, they contain oxygen on they lack oxygen on carbon number two. Then we have discussed the three major building up of the nucleotide basics, that is about okay, that is about the sugar group, the phosphate group, the nitrogenous base, and we have and step by step we have studied how we need them, why we need them, what is their purposes. So when it comes to the five carbon sugar, you need their carbon number three and carbon number five to for that case of attachment of the phosphate group. Then when phosphate group attaches to them, we can term them as phosphodiester bond. When it comes to the phosphate groups are needed to for the bond formation of the the sugars. It holds the sugar molecules together. The nitrogenous bases are available because they're building up different DNA, holding different structures together. Next point is that okay, we have learned different forms of DNA, which is form A, form B, and form Z. A and B both are right-handed. Z is left-handed side. Then about helicase, which is obviously the that breaks hydrogen bond. Then we have learned DNA polymerase that is responsible for binding up different nucleotides together. So I hope the lecture might have helped you. So see you in the next video. If you have liked the video, like the video, subscribe to the channel and share it with your beings. Thank you.